Hello everyone, I am Dinesh Woodby. In this video, let us continue with the study of gene transfer techniques. So in the previous sessions, we started this topic and we understood what is gene transfer and what are the different types of gene transfer techniques which are commonly used in genetic engineering. And in this video, let us concentrate on transformation and transfection methods of gene transfer technique. Transformation. As I already mentioned in a previous session, this is a process which naturally occurs in the bacteria. So, what is transformation? It is a gene transfer. Transformation is a process by which DNA is introduced into bacterial cells. So normally bacteria are capable of exchanging genetic material by means of transformation. So basically transformation is a technique where the DNA is transferred from a bacteria into another bacteria. So this is uh, transformation, and artificially this technique is widely used in genetic engineering. The steps involved in gene transfer transformation are selection of donor DNA. So the first step is to select the DNA sequence that will be transferred into the recipient cell. This donor DNA can come from a variety of sources such as another organism or synthesized in a laboratory. As I am repeating many times, the main aim of genetic engineering is to introduce a gene of interest from one organism into another organism where we want it to be expressed. So in the first step in transformation is selection of that desired gene or the donor DNA. Preparation of competent cells. So this is the next step where the recipient bacterial cells are made competent. That means the bacterial cells which are supposed to receive the DNA, they are made more susceptible to take foreign DNA. That means a normal bacterial cell will not take the DNA which is present outside. Now, by using some techniques like the chemical or electric techniques, we are going to make the bacterial cell competent so that it becomes more susceptible to uptake the DNA which is present outside the cell. So that is the second step. So this is where our gene transfer techniques are used. Then delivery of donor DNA. So during the gene transfer, the DNA gets inside the bacterial cell, and the mixture is subjected to a brief heat shock, electric pulse, or chemical treatment. So this is the process of gene transfer, where we use either a heat shock, electric pulse, or chemical treatment, and by the creation of temporary openings, the bacterial cell is going to take that DNA inside. And once the DNA enters into the bacterial cell, after the delivery, the bacterial cells are allowed to recover and grow in a suitable medium. The transformed bacterial cells can be identified by the expression of specific markers. So, uh, all the bacteria which are present in a medium will not take the gene of our interest. So we have to identify the bacterial cells which are transformed. So what is transformation? Transformation means the bacteria which has taken up the gene of our interest. The bacteria which has taken up the foreign DNA, that is a transformed bacteria. That's why we use the name transformation. So why it is called transformation? Because after taking up the foreign DNA, the bacterial cell gets transformed. So that's is the next step, where we are going to identify only the transformed bacterial cells, because that is the one, those are the ones which have our gene of interest. And then we can verify that by using different techniques, right? Like PCR, southern blotting, western blotting, or some antibiotic resistance methods, etc. So here, let us just refresh the same explanation here. In this example, say this is a human cell from which we are going to isolate the gene, that is a growth hormone gene. So this is the gene of our interest, right? So we are going to isolate it from a human cell because growth hormone genes are present in the human genome. And this is the bacterial cells from which we are going to obtain the plasmid. So you are already familiar with this concept. You already know that this plasmid is going to be used as a vector, right? And EcoR1 is the endonuclease enzyme which is used to cut the human DNA. The same EcoR1 endonuclease enzyme is to cut this vector. And because the same endonuclease enzymes are used, it will produce the same type of sticky ends in both vector and the gene of our interest or donor DNA. So this is the DNA which is having sticky ends, and this is the vector which is having sticky ends. Now because of the sticky ends, this DNA will get incorporated, will become part of this uh, structure. And now we have, when these two get combined with the help of ligase enzyme, now we have a recombinant DNA. So what is this recombinant DNA? This is a recombinant DNA which is made up of a plasmid with the gene for human growth hormone. So this recombinant DNA, by using different techniques like transformation technique, it is inserted into the bacteria. So using transformation technique, we are going to insert this recombinant DNA into the target bacterial cell. Now this is a transformed bacteria containing the human growth gene. Now technically speaking, this bacteria, because it has the gene for human growth hormone, in the culture medium, it starts producing the human growth hormone. So this is an application of genetic engineering, and it is also an example for transformation. Next, let us try to understand transfection. So what is transformation? Transformation is a method where you are going to introduce DNA into the bacteria. Then what is transfection? What is the difference between transformation and transfection? Transfection is the process of introduction of foreign DNA or gene into the eukaryotic cell. So this is the difference, right? In a transfection, we are going to introduce the gene of our interest into the eukaryotic cell like a plant cell or animal cell. Transfection can be done using either physical or chemical methods. So the same gene transfer techniques can be used for transfection. So remember, basically gene transfer techniques are same. So if you are transferring the DNA into the bacteria, the name you are going to use is transformation. And when the gene of our interest is being introduced into an eukaryotic cell, like a plant cell or animal cell, then we use the term transfection, right? So in the transfection, the techniques used are direct gene transfer techniques. So normally we use the direct gene transfer techniques. This gene transfer technology enables the study of gene function and protein expression in a cellular environment. So the end result or expectation, it depends upon the experiment. chemical transfection. So this method involves the use of chemical agents such as cationic lipids or polymers to form complexes with DNA or RNA molecules. These complexes can then be introduced into the cell through a variety of methods such as electroporation or microinjection. So, uh, chemical transfection is a method where we are going to mix the DNA or RNA with some chemical agents like cationic lipids or polymers. So what happens is the DNA or RNA molecule which is surrounded by these chemical agents, they are uh, they can be taken up very easily into the eukaryotic cells when we use a technique like electroporation or microinjection. So remember, directly transferring DNA is difficult. So they are going to use some chemical agents which helps in this transformation. I mean the transfer of DNA into the target cell, eukaryotic cell. So you are going to mix DNA or RNA molecule with suitable chemical agents. There are so many different type of chemical agents which are used, and in one of my previous sessions I mentioned the name of chemicals, right? So you can go through that. Electroporation. This method involves the application of an electric field to the cells which temporarily disrupt the cell membrane and allows the DNA or RNA to enter into the cell. So when the target cells are subjected to the electrical field, they form pores through which the DNA can enter into the cell. Microinjection is another technique where the DNA or RNA is physically and directly introduced into the cell using a fine needle, a microneedle. Viral transduction. This method involves the use of a virus as a vector to deliver the genetic material into the cells. So we already studied about the lambda phage viruses which are used for this transduction type of transfection or transfer of DNA. So here you can see this is a chemical transformation where calcium chloride is used for transfection. So using chemical method, the DNA of the interest is introduced into the cell, and it is a physical transformation where nanoparticles are used. As I already mentioned, so many techniques are available nowadays. So this is a technique where the nanoparticles are used through which using which the DNA is passing through the plasma membrane of the target cell. And this is electroporation. So due to the electrical charges, the plasma membrane becomes porous through which the DNA can enter. And combined transformation, so where both the nanoparticle or chemicals are used along with the electroporation. So here the chemicals or nanoparticles are used to cover the DNA, and then the electroporation is used. The electrical charges are used to create the pores. So the name itself tells you electroporation, the pore formation due to the electrical charges. And just to mention some other techniques like sonification, so ultrasonic sounds, different higher frequency sounds are used for the purpose of for transfer of DNA. So like this, there are different types of techniques which are available for the gene transfer for transferring the DNA into the eukaryotic cell. So here the same explanation, the pictures depicting chemical transformation and electroporation. So this is about. Next, let us see electroporation. I already explained what is electroporation. Let us just repeat the same. Electroporation is a simple and rapid procedure by which DNA can be transferred into cells. In this method, a high voltage pulse is applied to a suspension of cells. So in the media, we have the cells, the target cells, especially the eukaryotic cells like plant cells or animal cells, into which we want to transfer the gene of our interest. So those cells are subjected to a high voltage pulse, right? And DNA molecules which have to be transferred, they are also present in the solution. Now what happens? Due to the high voltage pulse, pores are formed in the plasma membrane of the target cells, and through these pores the DNA enters into the cells. Later the plasma membranes get resealed. So once the voltage is restored to a normal condition, the plasma membrane will get resealed and the DNA becomes part of the target cell genome and it will be expressed. This is a physical type of transfection method of gene transfer. So electroporation can be considered as a physical type of transfection because we are using voltage here. So here you can see this is a target cell and this is the intact plasma membrane, and these are the genes of our interest which we want them to enter into this cell. So now electric pulse is applied. When the electric pulse is applied, you can see the polarization here. So all the positive charges and all the negative charges accumulate here, and that results in the formation of pores. So now the temporary pores are created through which these DNA of our interest, the DNA molecules or genes, will enter into the cell. You can see here they have entered into the cell, and then when the conditions are restored, the plasma membrane is resealed. But you can see here many of the DNA are now part of the cell, and these DNA will be expressed and they will be used for further studies or applications. So that is the electroporation. So in this video we studied the transformation and transfection, and one of the transfection method we studied, that is electroporation. So these are the references you can use. So that's all for this video. Thank you.